Hello, this presentation is an introduction to marine mammal stranding networks. It is performed in the framework of the CARIMAM project, which is co-financed by the Interreg Caribbean program under the European Regional Development Fund. The CARIMAM project aims to strengthen the network of stakeholders involved in marine mammal conservation in the wider Caribbean region. One of the objectives is to strengthen capacities among the CARIMAM members. This presentation is the first of 4 dedicated to marine mammal stranding network strengthening. You will find here an introduction to stranding networks. A few words about your trainer, myself. I'm Claire Piusineri. I'm a marine biologist, and I've been working for the SPORAC, the Regional Activity Center of the Specially Protected Area and Wildlife Protocol of the Cartagena Convention since last year. I've also been member of the French Stranding Network for almost 20 years now, and I have been coordinating the strengthening of the stranding networks in 2 French outsea territories, Mayotte first in the Indian Ocean, and then French Guiana. Most of the material presented here is from the Marine Mammal Stranding Guide of the French Stranding Network. We will first see what is a stranding network, then we'll see why it is so valuable to set up a stranding network, then what data and samples can be collected, and then we'll see why stranding networks are long-term and wide-scale projects. So what is a stranding network? So first, what is a stranding? A marine mammal is said to be stranded when it is found on the shore, dead or alive, but unable to return to its natural habitat by its own means, because it is weakened or injured or it has a pathology, etc. There are 3 main types of strandings. First, individual strandings, which concern isolated individuals. Then multiple strandings, which concern a large number of individuals which may belong to the same species or not. And they are found over a short period of time, a few days to a few weeks, and they are located in a restricted geographical area. There are also mass strandings, which are simultaneous strandings of several cetaceans belonging to the same species and the same social unit. It mainly concerns gregarious odontocetes species, such as melon-headed whales or pilot whales. Stranding networks may also have to deal with animals at sea, both dead and alive, but in distress. Distressed animals are often trapped in coastal infrastructures or entangled in marine debris, such as fishing gears. The causes of strandings are multiple, but 3 main categories can be distinguished. First, natural causes, which can be diseases, predation, inter- or intra-specific competition, etc. Then you have anthropogenic causes, such as bycatch, which is the primary cause of strandings in many regions of the world, collision with vessels, acoustic pollution, oil spill, or ingestion of plastic, etc. And the third main cause of strandings is environmental, which can be climate change, storms, algal toxins, earthquakes, etc. So what stranding networks do? Well, they bring together scientists and volunteers and coordinate their response when an animal is found stranded on the coast or distressed at sea. So why is it so valuable to develop a stranding network? First of all, well-trained, structured and coordinated, stranding networks can achieve a lot for marine mammal conservation from little resources, both human, logistic and financial. Of course, their first purpose is to be in capacity of returning alive animals to the sea. But they also bring people together with trainings and fieldwork, which often results in the development of collaborative projects to enhance species knowledge and conservation. Of course, stranding networks also often involve volunteers at several steps of the process, such as alert, data and sample collection, or communication. And in that sense, it has all the virtues of citizen science, such as the education of the large public to conservation issues and building bridges between science and society. And of course, marine mammal strandings are often the main source of data and biological material to study these species and are an essential support to scientific research and public conservation policies. Indeed, the data and samples collected allow to reinforce knowledge on the biology and ecology of the species, but also on the threats. For example, when no dedicated survey can be conducted at sea because of limited logistic and funding, strandings can provide a first estimate of species diversity in a region. For example, the analysis of stranded marine mammal sightings along the Pacific and Caribbean coasts of Nicaragua from 2014 to 2021 have provided the first description of the cetacean community in the country. Stranding networks can also provide information on the biology and ecology of species. For example, the analysis of stomach contents collected on stranded animals has allowed to study the diet and feeding ecology of various species all around the world. They allowed to study the type of prey consumed, the main foraging habitats, the trophic levels, the trophic overlap and potential competitions with fisheries, etc. And there are many examples of such studies in the scientific literature. Histopathologic analysis of tissues and organs from stranded animals can also help to identify natural causes of mortality, such as brucella. For example, cetacean brucellosis in Costa Rica was investigated from 2004 to 2016 and showed that striped dolphin is highly susceptible of this disease and that many of the stranding events from this species in the country were due to brain infections, as recorded in other latitudes. Finally, data collected on stranded animals allow to identify anthropogenic causes of mortality. For example, in Venezuela, the review of cetacean stranding events from 1988 to 2013 showed that 36% of the strandings were due to interactions with fisheries and 6% was a result of ship strikes. What data and samples can be collected and for what purposes? From external examination of stranded animals, one can report, for example, marks of collisions with vessels or marks of fishing nets or of inter or intraspecific interactions, but also evidence of concussion, and of course one can determine the morphometry and the sex. These data are very valuable to study, for example, mortality causes or a length-age relationship. When external samplings can be performed, the teeth can be collected, for example, to study age and growth. Some skin sample can be collected to study genetic, and some blubber sample can be collected to study ecotoxicology and diet. When internal samplings can be performed, the stomach, for example, can be collected to study diet and foraging ecology. The kidney also can be collected to study contamination by heavy metals. Some blood sample can be performed to study histopathology. From the gonads, the reproduction status can be analyzed, and from the liver, the contamination by pollutants. And a last important thing to consider is that for stranding networks to be valuable, they have to be designed as long-term and wide-scale projects. The first reason for that is that in most cases, reaching an acceptable sample size to perform analysis can only be met after several years of data and sample acquisition. And in the case of small islands, such as we have in the Caribbean, this is often not enough, and collaborations have to be developed with other territories. I found an interesting example of that with the Philippines stranding network. So the country of Philippines represents an archipelago of more than 7,000 islands, and it is divided in 64 coastal provinces. Stranding data were informally recorded in the country since 1999, and a formal national stranding network was created in 2005. But they had to wait until 2010 to perform their first review of strandings from the 178 events recorded. This study allowed for the first time to estimate the species diversity in their waters, the most abundant species, and marine mammal hotspots. And they had to wait 5 more years to perform their first analysis of the samples collected on stranded animals. And the paper was published in, the first paper was published in 2015. Besides, only long-term data collection can highlight significant trends in ecological and biodemographic parameters and threats. As an example, the continuous collection of stranding data along the French coast for over 40 years has highlighted recurring peaks of common dolphin strandings since the end of the 80s on the Atlantic coast. The examination of stranded animals showed obvious signs of incidental bycatch in 75% of the cases of the stranded animals. These results showed evidence for the emergence of a major threat, bycatch in fishing nets. In addition, some analyses are only valuable on a large scale, such as genetic and ecotoxicology. As an example, in Brazil, the genetic analysis of samples collected on 2 humpback whales stranded along the Amazonian equatorial coast and their comparison with the haplotypes of other individuals has shown evidence that these animals belong to the species Southern Breeding Stock A. This led to hypothesize that the stock A is extending its range to the north or recolonizing its historical breeding area. Finally, Collaborations are essential to provide support for both logistic and expertise in case of an unusual event, such as the stranding of larger cetaceans or a mass stranding, when many hands are needed. However, a high level of involvement is needed for a wide-scale and long-term monitoring, such as long-term commitment and sustainable fundings, a coordination at the various levels, local, national, regional, a minimum level of capacity and resources in each involved territories, a consistency in data and samples collection and storage, and of course, networking tools should be developed, such as a mailing list, trainings, meetings, databases, and common study projects. Thank you very much for watching this presentation. I hope it has persuaded you that stranding networks are very valuable tools for the conservation of marine mammals. Here are all the references I have used to prepare the presentation if you are looking for more information.